The first step that a species takes to reproduce is cell replication. It is a form of cell division that forms two daughter cells. Cell replication creates new genetically identical individuals. The cell cycle is the life cycle of a cell involving growth, replication of DNA, and division to produce two identical daughter cells. A cell must replicate its DNA during a process called symphysis, so that each new daughter cell can receive a full set of chromosomes. This allows for mitosis to occur, where the nucleus is divided into two genetically identical nuclei. Meiosis is a process where daughter cells are not genetically identical. Meiosis is important for the continuity of species, as it is required for sexual reproduction and creating genetic variation, which helps with the adaptability of a species in changing environments. During meiosis, crossing over of chromosomes occurs by exchanging sections of DNA carrying some alleles, which forms new recombinant chromosomes with a set of alleles, not found in either parent. These chromosomes are made up of DNA, which I will now discuss the importance of. Today I'm going to be showing a model of how DNA and RNA work together to ensure continuity and variation in species. The first part here is the structure of a chromosome. A chromosome is a complex structure that is made up of DNA on the inside, which carries genetic information. The inside of DNA is a double helix, which is made up of nucleotides. Nucleotides are the chemical building blocks of DNA and RNA. DNA and RNA are made up of nitrogenous bases and a sugar phosphate backbone. Each consists of a phosphate group, a 5-carbon sugar, and one of the 4 nitrogen-containing bases of adenine, guanine, thymine, and cytosine. RNA is a single-stranded, and DNA has a double-stranded helix with complementary base pairings. In DNA, the bonds between nitrogenous bases are always the same, as you can see on this model in which color coding is used. These complementary base pairings are A and T, which is held together by 2 weak hydrogen bonds, and G and C, which is held together by 3 weak hydrogen bonds. These bases always attach to the sugars in the backbone, and there is always a phosphate between 2 sugars. Chromosomes carry genetic information as a unique set of genes inherited from the parents. A gene is a sequence of nucleotides along a DNA strand, with start and stop codons and other regulatory elements. That specifies a sequence of amino acids that are linked together to form a protein. The position of a gene on a chromosome is called a locus. There. Groups of 3 nucleotides are called triplets. The DNA copies into messenger RNA through the process of transcription. During the copying of DNA into RNA, the nitrogen base thymine then becomes uracil. The triplet is called a codon, where the codon codes for one amino acid and can also provide specific instructions such as start translation and stop translation. This here is a model of the messenger RNA strand, which moves from the nucleus to the cytoplasm. Inside the cytoplasm is a ribosome. The messenger RNA then attaches to it. This is the site where translation occurs, in which the messenger RNA is translated into a chain of amino acids. An anticodon is the 3 nucleotides on the transfer RNA molecule that join to the codons on messenger RNA by complementary base pairing. These anticodons are what code for specific amino acids. After the process of translation, amino acids are formed. A peptide bond is a bond between 2 amino acids. Many of these pairings of amino acids then form a polypeptide chain, as this is shown. These polypeptides then form proteins, which are essential for a cell and organism structure and function. Functions of proteins include structure and support, transport and storage, enzymes, antibodies, and messengers. The process of polypeptide synthesis is a major influence in ensuring continuity in species, as it forms proteins which are essential for the structure and function of a species. Along with this process, there are also many other processes such as mitosis and meiosis, which create natural variation in a species, allowing it to have the mechanisms of surviving in changing environments, therefore ensuring the continuity of species. Thanks.